Welcome back, everyone. I hope you enjoyed your lunch break and the sun outside in Istanbul. I'm really excited for the afternoon sessions, and in particular the sessions on epigenomics in cancer. Before I introduce our next keynote speaker, I would like to say a word to my co-chair, the so-called problem child. I think he did a really good job in, I have to admit, he did most of the heavy lifting of organizing this workshop, sometimes through quite some trouble. So he used a somewhat longer break in the morning, so that was quite useful to get something for you. You were told, so we had the option between something wacky, 3 different options. One was no, the other one was beautiful, and this one is beautiful, beautiful. So I hope you enjoy it. Thank you very much. With that, I would like to now. You may, but I think you would miss something out on the fantastic speakers that are going to come. Also, we have a surprise, of course, at the end of the session. The last session, we will have a poster prize and paper prize to give, so do stick around. Very exciting news. And with that, finally, this is our last keynote speaker, I'm really excited to have him here. We had some overlap in Tübingen before, and you're now in Milan, and we're really looking forward to it. Thank you.
Thanks a lot, Gabriele, for the introduction, and thanks also, Giulio. He's already getting drunk, for both of the organizers for inviting me here and giving me the opportunity to discuss our recent work on the identification of epigenetic driver genes. So the situation that we are starting with is this observation that really, if you look at DNA methylation profiles of tumors on healthy, and so here just showing a heat map where I basically just took the top 500 most variable genes in terms of DNA methylation and the promoter sequencing and the sequences, and then just basically do a very simple dendrogram here. And what you can see is that there's really massive changes in the promoter sequences in terms of DNA methylation. And so what we are wondering is really like how does all these changes contribute to cancer progression? And I don't reiterate now Trevor's joke about the Ryan Gosling, but you can guess what we are aiming at, identifying the drivers of tumor evolution in terms of epigenetic changes. So the thing is, obviously, we have changes on many different levels of the cancer epigenome. I will talk in my presentation mostly now on DNA methylation changes, but we have really on all the different levels. We have levels on the level of histone marks. We see changes between healthy and tumors and along the tumor evolutionary trajectory. Top level chromatin organization changes. And as I said, I focus now in my presentation on DNA methylation changes. However, I will argue that the methods and the tools we're developing could be in principle also applicable to other levels, and we have also some preliminary data to make this point. So I'm really embarrassed, but I will start with a very basic overview of kind of this textbook idea of what DNA methylation does in general and specifically in tumors. And so probably in every molecular biology conference they would kick me out for this. A, because it's so simple, and B, because partly this textbook knowledge is also a bit, I guess, there will be some questionable remarks that I'm doing here in their simplicity. So, so what the idea is, DNA methylation is a reversible attachment, and this reversible is important for the following part of my presentation, to the DNA, to cytosine, right? And so there is de novo methylation, which is mediated by the DNMT3 complex, and there is methylation maintenance, which is mediated by DNMT1. And then there's 2 ways of losing this methylation information. There's a passive mechanism, which is just basically means what it says, so you can lose mostly during cell division methylation information. And there's an active demethylation process, which is mediated by TET, which is much less understood than the methylation de novo or maintenance. Okay? So this very, very simplistic idea of what it does to gene expression is that you might have a gene where in the promoter sequence you have a region that is hypothetically can undergo methylation, and if it's not, transcription factors can bind there and modulate transcription. While when it's methylated, the transcription factor will not be able to bind, and this leads, in most cases, and this most we will discuss in a second, will lead to the close by gene being switched off. Okay, so how does this can this potentially contribute to cancer? Obviously, so we could think of a normal cell having a transcription factor binding in front of a tumor suppressor gene, and then this gene is being transcribed, or at least it's inducible, and then in cancer the promoter sequence can get methylated, and so the tumor suppressor would be silenced. Obviously, the same model works the other way around, that you could have oncogene that is transcriptionally silenced in the normal cell and then gets activated through demethylation. Okay? So the three questions I'm going to address today is the first one is which of the many DNA methylation changes contribute to cancer, and what I will describe in this first part, and this will be the longest part or the main part of my presentation, is how we basically use tools of population genetics to identify genes where the level of hypermethylation or hypomethylation is surprising. And so basically we develop statistical methods of asking what is the surprise here, right? And the second is the second part of the presentation, which will be rather brief. I will ask if the promoter definitions we have, which are mostly static in the sense that people typically apply either constant length definitions of promoters or use the encode, whatever, saying this is here, this is kind of this length or that length. But I will ask if this really helps us to understand best the effect of DNA methylation in cancer. Okay, and the third one, I will take the liberty of doing a little bit of a detour where I will ask how network approaches can help us to identify modules of basically DNA methylation cancer drivers. Okay, and I would ask you really, if there's something unclear, please interrupt me, and I prefer to discuss with you in the middle of the talk if there's something, because I will touch on different topics here, and I prefer to not finish than leaving you here a bit puzzled what this guy actually wanted to say. Okay, so let me start with the first part of the presentation. So again, I showed you in the beginning this heat map, which I wanted to use to illustrate that there's a lot of differentially methylated genes. So if you use really very basic statistics, you have now your whatever, let's say melanoma methylome, and then you basically pull the information of DNA methylation by promoter sequences, let's say 2 kb, and then you ask, so do a Wilcoxon test or whatever you want, and then you ask how many differentially methylated genes I get. You get thousands, okay? Like around like one quarter to 1/5 of all genes show signals of differential methylation. And this is obviously makes it a bit difficult to understand how these many genes contribute to cancer, right? And even more, like very few of these genes have a described role in cancer. So if you take now the known oncogenes by whatever method, right? Could be frequently mutated, could be something that has been characterized in the literature, and then you ask how many of my several thousand genes have a described role in cancer. Is very little. And the other way around, and this is shown here on the right, if you take now all your oncogenes or tumor suppressants, which fraction gets in any cancer type differentially methylated, again, you end up with a very small fraction. So it's not so easy to understand how all these differential methylation events really contribute to cancer, okay? Even more, the problem is that many of these hypermethylated genes, so which under this kind of textbook assumption of that methylation is mostly transcriptional, leads to transcriptional silencing, many of the genes which are hypermethylated are already not expressed in the healthy tissue of origin. So raising the question, why should they get hypermethylated, right? So questioning if there's selection acting on these genes to get hypermethylated and silenced. And then many of these hypermethylated genes are also olfactory receptors. And I'm mentioning this here because we were like in a bit similar situation 10 years ago when the first tumor exomes, or let's say 15 years ago, the first like large cohorts of tumor exomes come out where people did like naive statistics on point mutation data, and then the strongest enrichment typically was olfactory receptors, which we believe now is an artifact. Okay, I'll come back to this artifact character of these olfactory receptors. And then last, and this is maybe a bit more complicated to digest, but methylation changes, this is something we already published a few years ago, and other labs were having similar observations around the same time, is that actually methylation change in aging mirrors closely methylation change in cancer. And what we think is that this is not because obviously there is a relationship, no, that aging is the biggest risk factor for cancer, but what we think is more likely that there are kind of common underlying, let's call them for now, confounders that explain the similarity in cancer and aging. So let me start. If we look now here, this is a cohort of melanoma patients where we have basically we just compute the ratio of methylation level in tumors versus the methylation level in cancer. So we have control tissues. This is actually from TCGA. When I say we, so the first analysis mostly TCGA. I'll come to the data later. I mean publicly available data. And what you see is that really along the cancer methylome, epigenome, no, you have these peaks and you have these valleys. No? And again, some of you might feel reminded to the observation that for point mutation data, we have very similar profiles. It's again here, one chromosome arm from melanoma patients. But again, we have this black line here denotes local point mutation rate for melanoma patient binned by on a megabase window scale. Okay, and there's two, let's say three take-home messages from this plot, which is from a paper from, I think the reference comes in a second, from Bas Polak from 2015. And the three take-home messages are, the first one, we have really strong variations, no? along the in this case genome. Second, there's a second line which relatively closely resembles the black line, which is the blue line, and the blue line denotes chromatin compaction. So there are covariates, genomic or epigenomic covariates, which explain the amount of mutations you can expect in a certain region, right? And the third take-home message is, this is a problem if you do naive statistic, because you could have two genes, a gene A and a gene B, and the gene A, let's say, it sits here in this mutational valley, and a gene B that sits here in this mutational peak region. And if you compare the mutation frequency between these two genes, if you take whatever 100 cancer patients, the exome, you will assume, wow, this gene is related to cancer, right? Given the background of elevated mutation rates in cancer, you will see, okay, this gene likely is a potential driver versus is not, right? And now the clue is that most of these olfactory receptors sit in this closed chromatin regions, so they appear as mutational drivers, but they aren't, okay? And just because this motivates now what we are doing for DNA methylation information, how people solved this for mutation information is, let's say, you can roughly speaking divide this into two approaches. The first one is using these covariates, okay, and ask, can I predict for each of each window, each gene location, the expected mutation rate, and then I basically just factor this out or normalize my observed mutation rate by the predicted one. Okay, so and what you see here is this is again from this Polak et al. Paper. What you see is that really you can predict quite well the local mutation rate. So you can use these covariates to factor out the expected mutation rate when you have your observed mutation rate. And the second approach, Trevor mentioned this yesterday, is the DNDS, where you use mutations where you assume that they evolve selectively neutral, and this is not always true, but probably it's a safe assumption that in this case synonymous mutations do have lesser impact on tumor progression because they have lesser impact on protein function, right? So synonymous mutations being mutations that do not change the amino acid sequence. So, and then you can basically normalize your observed rate of mutations by here the expected one, the proxy for the expected one is the synonymous mutation rate. Okay? So, now, how can we apply these ideas to DNA methylation information? And for DNA methylation, it's very difficult to define selectively neutral changes. So this DNDS thing is difficult to implement. So what about covariates? And the first question obviously is, are there covariates? Huh? And so this is what we tested first. And so the data we were using is mostly DNA methylation, publicly available DNA methylation information from TCGA. But we also included 2 other cohorts, which I mentioned here because they have 2 important features. First is whole genome bisulfite sequencing information. So we have a much higher coverage. You basically have full whole genome coverage of methylation level of CpG sites, versus this Illumina arrays that were used in TCGA have a strong restriction to basically like 500,000 sites. And the second fact that we're exploiting from this other 2, sorry, it says 3, it should be 2. Was a test if you are if you're awake. And the other 2 cohorts, the second feature we have from the same patients, we have whole genome bisulfite sequencing information and RNA-seq data, which will become important a little bit later into my presentation. So the first question is, are there genomic or epigenomic covariates of DNA methylation change? And we did the same thing as before, but basically here, so also on megabase bins, and we asked basically what are epigenomic features that are correlated with DNA methylation change. And now the important piece of information, and I will reiterate this a few times, is that this is information we get from basically the healthy cell, the tissue of origin, and that predicts basically what happens in tumors. So it's not this kind of trivial relationship I'm describing here and in the following slides, that we look at the tumor and we see DNA methylation change is correlated with something which is also happening in the tumor. It's really, and sorry for again being very basic here, but what we see here is that some properties of basically other cells which are not transformed cells predict what happens in a tumor. And you see that here, for example, for replication timing data, so this is basically the strongest covariate that we and others identified so far. You see a very good correlation between basically if a region is early or late replicated and the DNA methylation change from healthy to tumor. Okay, so genomic or epigenomic covariates exist. Now let's make them actionable to normalize our observed DNA methylation change signals. Oh, not yet. Just one little comment. Just it's more like kind of this is irrelevant to what I'm doing in the next slide. But just I want, yes.
Can you just say what's on the x-axis, the scale? So it's 0 is on the right side, right?
Yeah, so it's mostly loss. It's mostly loss, yeah.
So the high methylation is in late replication.
So there's a lot of methylation loss in late replicating regions, and this is exactly related to this. This happens in a cell division. So basically, in a cell division dependent manner. So cell types with a higher cell division rate have a stronger loss of DNA methylation in the late replicating regions, and we think this is because probably this is here this this passive DNA demethylation mechanism taking place. So that probably during replication, in later replicating regions, the copy machinery becomes more sloppy. Probably DNMT1 sits in the early replicating regions, and in the late it doesn't properly transfer the methylation information. This is our idea about this, but this is not relevant for the question how to deal with it when we want to identify these methylation driver genes. So what we did is that we took from the roadmap epigenomics, because the advantage is here that we have really for a large fraction of the tissues for which we have cancer DNA methylation data, we have also from the healthy tissue of origin epigenomics data. And so what we do is we basically we don't train any complicated model. We just fit basically linear models, and we ask how these epigenetic features explain the DNA methylation change we observe in different cancer types. Okay, and what you can probably only see until the first row is here is basically this important. This is here the y-axis is the explained variance, no? And so basically here is 0.2, here 0.4, and so we explain a reasonable, I would say, an average, let's say around 0.3, 30% of the variance with our already very simple linear models. Okay? And so the features, we can also ask then what are the important features, so and so on. We find H3K27 trimethylation and some features which really seem to be predictive, either on individual promoters or megabase scale. So we tried this a bit like varying the scale, but the picture is always the same. So it seems to be predictive of methylation change. Again, and this is the last time I remind you, this is the healthy tissue of origin, which predicts here what happens in the tumor. So now we use an approach that was actually, the idea was borrowed from Lawrence et al., which did something very similar. This was one of the first approaches, this covariate basically approaches for dealing with this local variation in mutation rate 10 years ago. And so we do a very similar algorithmic approach. What we are doing is we are basically now projecting every promoter into an epigenetic feature space. Okay, so we have this 827 dimensions of our roadmap dimensions, and we project every dot here as a promoter. And let's say we are interested in this red promoter, and we want to know if this DNA methylation change we observe for this specific promoter is surprising or not. Okay, what we can do is we can define a neighborhood in this covariate space and can ask what is the change of these black promoters here. Okay, and then just basically create a background distribution of expected DNA methylation change and ask how likely is our observed DNA methylation change under this background methylation change distribution. Okay, and this is what we basically implemented. And what we see, so obviously this was a toy model. This is the real data. And I think this is really beautiful because I don't know, again, if you see it behind the second row, but if you want to come to the front just for this plot, because I think this is the most beautiful plot. Sorry for already being a bit, because really what you can see here is that in this epigenetic feature space, basically color encoded is now, so this PCA of the epigenetic feature space, and color encoded is a methylation at the increase or decrease. No? And you can really see that methylation in and decrease cluster by basically in this feature space. So you have this basically here, this methylation increase region, and we have the methylation decrease region. And obviously, if you now find a promoter which is like located here and increases, obviously it's not surprising. But if you find it here, no, it's surprising. So this is basically, in a nutshell, our way of defining DNA methylation drivers. So here's one example. I left out here the axis labels, but just to, well, the labels I could have put. But basically here on it's the same plot as before. So it's basically DNA methylation change on the x-axis, and we have here two genes which have exactly the same level of DNA methylation change. One is APC, and the other one is HOXY11. APC, you probably know, is a famous driver of colon cancer. Yes, Trevor? So we did it with and without, and it doesn't make a difference. But of course, all these features are also quite correlated with each other. But again, it's the methylation of the healthy tissue of origin, right? So what you see is that basically you have the same level of DNA methylation change for these two genes, but in one case it's surprising given the background. In one case, it's actually not surprising given the background. So we would consider this a gene which is a good candidate for being really selected for DNA methylation change, whereas this is not. So if we apply this type of correction, it's not really just a correction because we identify also genes which were not in the original set of differentially methylated genes, because some really the methylation change is so small that you would not detect it with a standard statistical method, but yeah.
Basic question: how do you define methylated genes, for example? Is there just a window around the promoter?
Yeah. So in this case, and this is perfect, because this will be the transition to my second part. So in the first we took static definitions of 2 kb upstream. I think 200 downstream and 2 kb upstream of the transcription start site. But I come in a second that this is actually I would trash my own work in the second part of the project. But well, not trash. We recover with also alternative promoter definitions most of what we see here. So so we have here again, like you have on the y-axis the number of hypermethylated genes, and this is only hypermethylated. You find several thousand, and then you apply this kind of approach of identifying methylation drivers, and you end up with a few hundreds. The next we ask, can we estimate the number of drivers on a patient-specific level? And this is, we did it quite naively, so there's some limitations to this approach. We basically just ask, so now we have our cancer type specific driver list, and then we go back to the naive statistic and then ask basically how many of them really change their methylation change in a specific patient. And what you can appreciate here is that like around 50% of the DNA methylation driver in a specific cancer type are really differentially methylated in each individual patient. Okay, of course there is a large variation, but this is like as a rule of thumb. There's a relatively high agreement between the tool set of methylation available with the methylation drivers and what is really methylated, at least higher as for point mutations. So then we ask, do these methylation drivers make sense? So first we ask, because I explained to you the problem that many of the genes are not expressed in the healthy tissue of origin that are differentially methylated then in the tumor, or hypermethylated in the tumor specifically. So now I'm focusing on hypermethylated genes. And we see that the light blue is basically uncorrected, dark blue is corrected method driver, which I call the two, but I'm typically refer here to with just corrected, uncorrected for simplicity. So uncorrected, you have basically an enrichment typically of non-expressed genes. Yes?
Can you also reduce that list of unexpressed genes by using a correlate measurement like 27 acetylation to identify, or 27 acetylation or 8-form trimethylation identify the active promoters in a.
If we reduce a set of non-expressed genes, why would we?
Well, so from what I understand, part of the problem here is that you're looking at methylated promoters throughout the genome. You're trying to identify the genes that are active in those tissue types. But can you use histone modifications like K-form trimethylation that mark active promoters in that tissue type?
No. We do it on a tissue-specific level, obviously. So we define for each tissue a specific set of non-expressed genes, but we do not take into account. But it's a fair point. To be more stringent, one could check if basically it has certain properties, epigenomic properties in the hedge. We should check this as well. Yeah, we didn't do it here. So he is really just saying, okay, the skin, this is not expressed, and what happens on methylation level. And so then you can see that we reduce largely, the number of. So we have typically like a significant depletion of non-expressed genes after the correction before we have an increase. So what about tumor suppressor genes? Obviously, so this is we tried this with two sets basically of tumor suppressor gene definitions. One based on recurrent mutations, and the other one a literature set. The picture looks always similar, but just of course as a disclaimer, obviously tumor suppressor mutation as tumor suppressors is not like a gold standard set or a ground truth set to validate here. No? You would not expect 100% agreement, but just to use as a proxy of something that makes sense. No? And what you see here is that basically the uncorrected set. So here's random expectation, and you see that you increase largely the overlap with tumor suppressor genes once you apply this correction. So I will come back to the tumor suppressors in a second. Just very briefly, we compared with other methods that are out there. None of them. There's one relatively recent addition which also uses replication timing as a covariate, which is so most of them, they use other, so two of these methods basically use, mostly rely on expression information. MASTIC also includes expression information, and then there are two which are basically just using a bit more sophisticated statistical approaches, but without using additional, let's say, information to classify DNA methylation driver. And what you can see here, if you use this tumor suppressor gene, in 9 out of 13 cancer types we perform well, but there are also other tools which might be in single cancer types superior. For example, I think an interesting case is in thyroid cancer. I don't want to go now 10 slides, but I don't know if you remember. Thyroid cancer is a cancer type, the cancer type where we really predict the least of the variation in DNA methylation with our simple linear models, and this is also the one where we perform worst in all types of evaluation criteria we have, right? And the other tools outperform us there. So if you would ask me if you want to interpret your thyroid cancer epigenome, take another tool for colon and lung, I think we do quite well. Interestingly, I just don't want to go into detail anyway. You can, I'm sorry, probably it's difficult for you to see that plot, so you have to trust me and Julio, who can still probably see it. So interestingly, we perform as well as tools which are incorporating expression information. No? So the three of the tools I mentioned, no, they directly ask basically, are genes really down-regulated as a consequence of methylation increase or vice versa? And we without using this information. No? I mean, you could argue again, we use epigenetic information which is predictive of survival, but again, we use it only from the healthy tissue of origin. We perform as well as basically as tools which directly in terms of kicking out genes which are non-expressed or increasing the overlap with differentially expressed genes. So just briefly, to discuss a little bit the function of the genes with respect of tumor suppressors. So we ask if we now overlap this potential DNA methylation driver gene list from different cancer types, what genes are showing up in more than one tissue? And so in this plot, basically, I just show the number of cancer types in which this gene is in this potential driver list. Okay, so we have here on the very right as large as the 9 cancer types here, 8, 7, 6, and so on. And then here I mark just a few. So the gene which is really popping up in almost every cancer type is MLH1, which is nice because MLH1 is really like, it's a known tumor suppressor, which is also known to be epigenetically silenced at least in colon cancer. APC is occurring in relatively many cancer types, and this APC is also interesting because what I'm comparing here on the right in this needle plot is the mutation frequency versus the DNA methylation inactivation frequency. And what you can see here is that in colon cancer, really APC sticks out. No? So you have like 70–80% of all colon cancers have an inactivating APC mutation, whereas in other cancer types, so in red is basically hypermethylation silencing. Sorry, there's a typo here. It should be hypermethylation, not hypermutated. So in red, and you see a much more uniform distribution. No? So there's many more. What we claim is there's more cancer types in which APC is silenced, but by other mechanisms than in colon cancer. Okay, so. Still, we were not very pleased with just doing simple functional enrichment analysis because in most of our cohorts, we got only few significant Go terms. They usually made sense, but we didn't see really striking patterns that would explain the biology behind these genes being selected for being silenced by methylation. The strongest signal we got, and this was consistent for the bisulfite sequencing data and the array data, was an enrichment for the glutathione pathway in prostate cancer. So we looked a little bit more into this. And glutathione pathway has a very important role for cellular detoxification and protection protection and detection of mutation burden in cells, right? And so GSTP1 was the gene where we had the most strongest signal actually in several cancer types, but we focused here on prostate cancer. And so here's just like the plot you saw before. So we have really a huge distance of the observed and the expected methylation change for this particular gene. And so we asked, is the methylation state correlated with the overall mutational burden, which is actually detected by or in theory should be detected by GSTP1. And what we see is that there's actually a pretty good correlation between GSTP1 methylation state and overall mutation counts in prostate cancer patients. And this is not something that you have overall a correlation. Could be that there's some confounder that, I don't know, a lot of methylation increases mutation rate, etc. And this is not the case. So GSTP1 is among the top 10 genes where you see this correlation and the methylation state of the specific gene in the overall mutation signal. So this is the end of the first part of my presentation. So what I tried to convince you of is that we have a regional variation in DNA methylation rate, and the approach that we address this, and there might be other approaches in the future, but how we did it is like really trying to incorporate as many epigenetic features as possible from healthy tissues of origin, and then asking how surprising this methylation change is. Okay, so I have around 15 minutes more, no? So 10, 15 minutes. Okay, good. So the other two topics will be a bit, yeah.
With the MLH1 methylation change, does that correlate with mutation rate?
I don't remember.
Right.
So, yeah, because I mean, you would expect, right? Yeah. No, I don't remember now.
Also, mismatch pairs only present in very small.
Also for APC, probably you could, no. Well, MLH1 should be much more direct. I need to check. I don't even remember if in huh? Sorry?
He had hypermutated, but he meant hypermethylation.
No, on the slide before it was the top.
Yeah. No, both are right. Yeah, I mislabeled one plot, but also it's valid to check this. But I don't remember now if we only did this type of analysis in prostate. I don't know if MLH1 popped up in prostate, but there is no reason to not do the same analysis in any cancer type. It's a good point. Yeah, we didn't check this, but it's good thing to do. Yeah.
Can I have one
Yeah.
question between as well? Can I just wonder how much is the methylation changes actually driven by sequence changes? So I mean, if you have a C and you have a variation of C, then you lose the methylation obviously. Or if you're having an alteration towards a C, you might methylate it. So have you looked at the correlation between sequence variants and methylation?
So yes. So I mean, there is a link, obviously, as you say, between methylation and so we checked in the next part of the project. But this is I'm not saying this to because it's true. We already here. I mean, it can be also. I'm always a bit afraid of this because this can be also confounder, right? And we observed now, and this comes now. I show a plot where we show that in specific region of high methylation change, we see also high mutation counts, and we try to apply several types of analysis to exclude that there's some confounding effect either in the same sample that we observe something as a methylation which is actually a mutation, or just that there's simply hypermethylated tumors can gain more mutations or anything like this. I think it's that these results are likely not driven by this confounder, but this is something we started looking a little bit into it, and we have the impression that this is not largely affecting. I don't have any plots here now to show this, but we for example, we exclude that there are systematic mutation patterns that you would, I think, expect if this was confounded the signal, that you would see specific types of mutations, right? Only C to T.
Well, no, somewhat related. The tumor suppressors are on like 2 hits. You have to knock out both alleles. Do you see this, that one is methylated, one's mutated?
Yeah, we tested this, but we did not see a strong signal for this. We did not see that, for example, we were surprised that, for example, with APC, So we thought, so because we basically see an 80% differential methylation and an 80% mutation, so we asked if this is somehow, if there's some statistical link, but there isn't. But yeah, would have been nicer if there was. Okay, let me come to the second part of the presentation. So I already mentioned it. Yes. No, it's still okay. The third part is, thank you. So what we observed is that there is typically a non-even distribution of DNA methylation change about what we consider a promoter sequence. What I'm saying now when I say we, I really mean we, not you. So because everyone has a different concept of promoter. So we started with first 2, then we expanded to 5. And what you see here is like basically on the y-axis DNA methylation change, and here on the x-axis is distance to the transcription start site. So here is 0, and here is -5, and here's +5 kb. Okay, and you can see that many, this is like one example, but many of the promoters, they look like this. There are certain regions where you have a strong methylation change, and other regions where you might have the opposing change of promoters and of methylation. And now here I show 5 static. This means not like ENCODE way, but ENCODE we have the same problem. I'm just using this here because it's easier to do statistics here. So basically 5 static definitions of promoters that we found in recent literature of people who did similar things as us. So trying to understand better the effect of DNA methylation on tumor evolution. And so here basically the length of this bar basically encodes the region that is considered a promoter sequence. And what you see here is basically you get opposing signals of DNA methylation change depending over which interval you average your DNA methylation change. No? So in some cases you find if you take a long definition, you would capture this entire peak. You say this promoter is gaining methylation. If you take a shorter definition, you will assume it's losing methylation. Okay. So we wondered how we can address this, and here just it's slightly more complicated because I'm mixing here 2 types of information which are important. So the y-axis changes now. The y-axis now is incorporating expression information. So what we did is we asked, is there some sort of agnostic way of defining these regions that we saw here, right, where we have a relatively consistent change of DNA methylation across in a spatially confined region of the promoter without using the DNA methylation signal, right? So and what we observed is that we often find clusters of region which have a consistent effect on nearby transcription. And so for this now we use these other cohorts I mentioned where we had whole genome bisulfite sequencing data and RNA-seq data from the same patient. And what we did, so we had both, we had for healthy control and we had for patients. And so what we did is we correlated each single CpG site, the methylation state, with the nearby transcription, okay? And what you see here, forget the color first. You see here's 2 clusters with consistent effect on transcription. So this is our friend GSTP1 again. So, and you see that here's one cluster where the CpG sites are positively correlated with transcription, and here's a cluster where CpG sites are negatively correlated with transcription. And if you now superimpose here by color encoding the methylation change in cancer, you see that they consistently are losing methylation while they are gaining methylation. Okay? So what we did now is that we came up with basically it's a clustering algorithm. I don't want to go too much into the details, but where we basically spatially and in terms of correlation on the y-axis cluster basically these CpG sites and ask, are there any close by CpG sites with a consistent effect on transcription? Okay? So with this we end up with a list of not promoters anymore, but just regions which somehow behave in a consistent manner in a way of a potential regulatory role on transcription of nearby transcripts. Okay? So with this we found 8,600 something negatively correlated, high confidence clusters and 3,400 positive, and around 5,000 genes. Interestingly, many of them having both positive and negative clusters. So just as a disclaimer, this is some very recent work, so we're still working on this. I just want to, it's more like, also like the third part, I want a bit question here the kind of the definitions we are using for that we take as granted, and not provide it now here already a complete story, but really showing something that we are working on like really this week. Okay, so excuse me if I cannot answer every question or leave you with some still doubts about what we are doing here. But anyway, so this is not surprising. This is just like a reconfirmation that what we are doing is producing what it should doing. Now we averaged again about our different promoter definition, the average basically methylation signal, and then correlated this with the expression. And what you see is, so basically for each of this, so each is, except these are clusters, these are basically promoter definitions, and you see what you would expect, right? So you have always a little bit left-shifted correlation with expression, no, for all the different promoter definitions. But with the clusters, you find this two concise sets, no, of positive and negative correlations. This is not surprising because we selected these clusters to have this property. No? Now it becomes interesting, because what happens is, if we compare healthy to tumor, we see that the healthy tissue, they're dominated really by negative correlations. A little bit against this textbook idea, they have a slight tendency to appear here as a transcription start site, so to appear downstream of the transcription start site. But suddenly, what happens in tumors is that you get this really like bimodal distribution of positively regulated clusters, correlated clusters, around the transcription start sites. So we obviously ask now, what are the properties of this negative and positive clusters? And the first observation we had, which was, I think, nice and confirming, is that the negative clusters do what you probably would expect them to do. They enriched a transcription factor binding sites, right? So this is again this textbook idea that if you have like the CPG islands of which they behave consistent, because probably they really block some transcription factors. And if we can compare to, we did different approaches compared to random clusters of basically non-correlated CPG sites, or just all regions outside the clusters, or the positive ones. So you find enrichment of transcription factors. Okay? And the second property is, and this is in the direction of what Gabriele just asked, we ask, are these clusters enriched in mutations? And this really, you should probably take as with a grain of salt, because we tried already to factor out now here potential confounders by asking if there's any enrichment of certain mutational signatures, et cetera. But we see also enrichment of mutations which we think are of regulatory nature. Okay, but this is something we still need to confirm. We're looking now also on the effect of transcription. We don't have enough mutations per cluster, so this is all clustered pool to make really statistics about wild type versus mutated clusters. But we see there a signal also with respect to effect on transcription. So what are the positive clusters doing? And here our observation is they are not enriched in the properties I was explaining before, but what they are enriched in is they tend to occur in repetitive elements. So many of them are also transcribed and retrotransposons, etc. So we think that this positive correlation probably is some sort of indirect mechanism, but this is something we still try to sort out. Obviously we ran our method driver tool on now this redefined regulatory regions. So this our correction method driver. And interestingly, we find that like really in these clusters, we have an even higher fraction of variance of DNA methylation that is explained by the epigenetic features of the healthy tissue of origin. Basically, so after applying the correction, we end up with a very small set of clusters that shows strong signals of selection, 28 clusters only, including GSTB1 that we found before. And this time we find a relatively strong enrichment for prostatic bud formation, gene ontology enrichment, which we think is promising given that we are looking actually at prostate cancer data. I guess I'm running out of time, right? So I should not enter the third.
Well, we have had a lot of questions already, so we have 8 minutes left this question.
I mean, we could also ask now if there are questions to the second part, and then if there's time, I can briefly move to the networks.
I don't know if I understood this properly, but these clusters are within the static promoter regions or anywhere in the genome?
No, so we limit it just for multiple testing regions to 5 kb.
But the clusters should correlate very, very strongly with the CPG islands or with
Yeah.
actually hypermethylated regions in healthy tissues
Yeah.
versus hypermethylated
Yeah.
bordering regions, right?
Yeah. So actually also the positives are enriched in this. I mean, by definition, because we spatially cluster, so what is annotated as a CPG island will also more easily pass our clustering filter.
Yeah, but if you're looking at like tissue-specific hypermethylated
Yeah.
regions in healthy tissues,
Yeah.
I mean, in these regions you basically can only gain methylation.
Yeah, yeah.
In the regions
Sure.
which are in healthy,
Sure,
in
sure.
the healthy tissue,
Sure.
100% methylated, you
Sure.
can only lose methylation.
Of course,
So
of course,
it's,
of course, of
it's
course.
kind of almost trivial correlation.
Yeah, no. I mean, of course, so you can only gain when. So this is limits, of course. So that we can only. So this clustering works only by the restriction of the properties of the. Of course, yeah. You're right.
So I had maybe slightly more high-level question, because I seem to remember a talk years ago by Schaefer, who was doing all of these experiments of checking whether things would bind methylated
Yeah.
DNA that would. So if you add methylation, does it abolish binding? And there were very, very few.
Yeah.
transcription factors.
Well, so there is actually we're using from Dirk and from some other studies. So they defined a set of DNA methylation sensitive transcription factors, and we see them enriched in
All right.
in
So
the binding
it's
sites,
only
which
these
in.
ones that are enriched.
So it depends, of course, how you ask the question, right? I mean, how much variation of transcriptional control that is explained by DNA methylation, or vice versa? If you take now these regions, is DNA methylation sensitive promoters important in these regions? So the second, yes. The first question, I don't know, but I would probably agree with Dirk Schubler that the more important thing. I mean, it's a sum of many different factors, and we're focusing on one aspect of a highly complex machinery, right?
Maybe following up on this question, has anyone ever looked at whether methylation is causing any switch from canonical to non-canonical transcription factors, or the ratios between them?
I'm not aware. I think would be a very interesting thing to do, but I'm not aware if someone did and could be interesting to do it.
When you did the clustering, did you try it out across different cancer types and find that the regions remain consistent across?
We have only whole genome bisulfite sequencing data to a sufficiently large cohort in prostate. We were applying now for access for 2 more cohorts where we could do this, but we don't have access yet. But yeah, we will do. But so for some reason, people did a lot of this in prostate. We need whole genome bisulfite in combination with RNA-seq. Okay. Okay, probably we skipped in the third. Or we do a vote. No.
You can go on for
No, but there's 3 questions here. So, no, but.
I'm sorry.
No.
Do enhancers also undergo a similar methylation change in time?
What's that?
Do enhancers also
Ah,
undergo a similar methylation
yeah.
change?
We started looking a little bit into enhancers. The problem is that it's much harder to associate them with genes, and then so you again have strong multiple testing issues. So we were a bit. I think it's very, very important to do, and I tried to convince my PhD student who was working on this to do it, but he's refusing it so far. But yes, I'm. Yeah, I tell him. I try again.
So do you think are there any influence from the copy number change? Because for example,
Yeah.
there is imprinted genes, and that is overlapping with the frequently copy alteration.
We did not consider this yet, but it's true this could, yes, it would probably make sense to see if any signals from copy number iteration might have an impact on what we observe, yeah, no, but we didn't do.
How often do you find the example where we show a group like the traditional annotation and then there's a positive and negative cluster?
So we find it more often than you would expect by chance. So in total there are, I think, like 800 genes where you have both a positive and a negatively correlated cluster. I'm at 2 minutes now.
No, I think you
Oh,
can go on.
well,
for
this
5
is
minutes
good.
if you want.
Okay, good. So then I quickly, so what, because I think this is nice, it goes a little bit